Bismillahirrahmanirrahim. In today's lecture, we are going to discuss an introduction to the endocrinology and endocrine system. We know that all of the body systems and organs coordinate with each other and their functions are dependent on each other. So, for making this coordination, various chemical messengers are used, through which, for example, one cell may communicate to the other cell. The types of communications are divided into those that are quick and short-lived. For example, the cytokines. The cytokines are chemical molecules, and we have studied them in detail while discussing inflammation, in which one cell releases a cytokine that may act on the same cell or on the nearby cells or the cells that are far apart. The cytokines may be interleukins. Interleukin means in between leukocytes, and they may be in the form of chemokines, which are responsible for chemotaxis. The second type of communication is made by the neurotransmitters. For example, the acetylcholine that is released by the nerve endings and act on the muscles to cause muscle contraction. There are some slow but sustained means of communication, and the most important uh, are the neuroendocrine hormones. For example, adrenaline, which is secreted by the nerve endings and acts on various body organs and tissues for various functions. Another example is the antidiuretic hormone or vasopressin that is produced by the supraoptic and paraventricular nuclei in the hypothalamus, and that is stored in the posterior pituitary gland, which is released upon signals. The fourth way of communication, and that is also a slow and sustained way of communication, are the endocrine hormones. Endocrine hormones are released by endocrine glands, and mostly they act on the organs that are far apart from the organ from where the hormone was released. The chemical messenger system in the body may act on autocrine, paracrine, or endocrine fashions. By autocrine, we mean that the cells that secrete a particular messenger act on the same cell. In paracrine fashion, they act on the nearby cells. And in the endocrine fashion, the messenger or hormone is carried in the blood, and it acts on a target organ that may be at a distance from the organ that secretes the endocrine hormones. Usually, the secretion of the endocrine hormones, is regulated by the feedback stimulation or feedback inhibition. When the blood level reaches to a defined level, and if the desired function is achieved, then the serum level of the hormone will have a negative feedback effect on the organ that secretes the hormones. And in this way, a defined and optimum level of the hormones are maintained in the body to maintain the homeostasis and optimal efficiency of the body systems and tissues. Here we take an example of the pituitary hormones. The hypothalamus, which is the major center located in the brain, The hypothalamus senses changes in the body, for example, changes in temperature, changes in the osmolality, fall in blood pressure, and other form of changes like injury or trauma. Then the hypothalamus, according to the need, will release certain factors. These factors are mostly stimulatory factors, but some are inhibitory factors. For example, the dopamine is an inhibitory factor which will inhibit the secretion of prolactin from the anterior pituitary. Similarly, for growth hormone, there is also an inhibitory factor which will inhibit the secretion of the growth hormone. This releasing hormone from the hypothalamus is the hormone 1. For example, for corticotropic hormone, corticotropin releasing hormone secreted by the hypothalamus is hormone number 1. Now, CRH or corticotropin releasing hormone will act on the pituitary gland, and it will cause the pituitary gland to secrete hormone number 2, and that in response to corticotropin releasing hormone, the hormone number 2 will be adrenocorticotropic hormone. Adrenocorticotropic hormone or ACTH is hormone number 2. Now, this hormone number 2 or ACTH will act on the adrenal glands, which are the target glands, and it will cause the adrenal glands to secrete the hormone number 3. For example, it will cause the secretion of cortisol. So, this hypothalamic pituitary adrenal axis is a way by which the body or the hypothalamus, after sensing changes in the body, will order or it will cause the pituitary to release hormones, and the pituitary hormones then will act on the target organs. Mostly the hormones secreted by the target gland, for example cortisol, that is secreted by the adrenal gland. Now the blood level of the cortisol will act as a negative feedback. If no more cortisol is needed, then the cortisol level in the blood will inhibit the pituitary gland, as well as it will inhibit the hypothalamus to stop secreting CRH, corticotropin releasing hormone, from the hypothalamus, and adrenocorticotropic hormones ACTH from the pituitary. When the cortisol is needed, this negative feedback inhibition finishes and it releases the hypothalamus and pituitary to release CRH and ACTH to cause secretion of the cortisol. So the serum level of the hormone is maintained by the feedback loops. The hormones are classified into 3 major types. The proteins or polypeptides, which are made of amino acid chains, include the hormones of the anterior pituitary gland and posterior pituitary gland, and also the hormones secreted by the pancreas, like insulin and glucagon, and they are controlled by the blood level of the glucose. The insulin and glucagon and pancreatic hormone secretion is not under the pituitary directly. Similarly, the parathyroid hormone, which is associated with calcium and phosphate metabolism and their levels in the blood, is also not controlled by the pituitary, but it is the calcium level of the blood and phosphate level of the blood that will control secretion of the parathyroid hormone from the parathyroid gland, as the sugar controls the secretion of the insulin and glucagon from the pancreas. Some hormones are known as the steroid hormones, and they are derived from the cholesterol. Mostly these hormones are made by the adrenal cortex in the form of cortisol and aldosterone. Steroid hormones are also secreted from the ovaries in the form of estrogen and progesterone, and testosterone is also a steroid hormone which is the male sex hormone secreted by the testes. During pregnancy, the placenta also acts as an endocrine gland and it secretes estrogen and progesterones. There are some hormones that are neither proteins or polypeptides, neither steroid hormones, but they are amino acids and they are known as the tyrosine derivatives. The tyrosine derivatives mainly include the thyroid hormones, which are the thyroxine and triiodothyronine, and the hormone adrenaline or epinephrine and noradrenaline or norepinephrine that are released by the adrenal medulla or by the sympathetic nerve endings. The tyrosine derivatives may behave like proteins or polypeptides. For example, the adrenaline and non-adrenaline, they bind to the receptors on the cell membranes, or they may behave like steroid hormones like thyroxine that doesn't need a receptor in the membrane and directly passes through the membrane to bind to its receptors on the nucleus. So, for a hormone to perform its action, it will have to bind to a receptor on the target cell. Now, the hormone and receptor complex will then perform the required or desired action of the hormone. For some hormones, the receptors are located in the cell membrane phospholipid bilayer. For some hormones, the receptors are located in the cytoplasm, and for some hormones, the receptors are located in the nucleus. So, the proteins and polypeptide hormones, which are amino acid chains, and they include the hormones of the anterior and posterior pituitary glands, as well as the hormones of the pancreas like insulin and glucagon and the parathyroid hormones. So all these are proteins or polypeptides and they have their receptors in the cell membrane. Because these hormones, they are not soluble in fat, and they are however soluble in water, and for that reason they don't need any transport protein for them to be carried in the blood. They directly flow in the blood, but they cannot cross the cell membrane because they are not soluble in water. They are hydrophilic and they have to bind to a receptor in the cell membrane. Various receptors are located in the cell membrane. For example, G protein linked coupled hormone receptors, they will bind to the protein or polypeptide hormone, and this hormone receptor complex will cause further changes through various systems. For example, the second messenger system, and that will be activated, and ultimately the second messenger system activation will lead to the nucleus and expression of various genes that are needed for the synthesis of structural and functional proteins and to achieve the desired function. So the proteins and polypeptide hormones for which the receptors are located in the cell nucleus, actually these hormones themselves doesn't enter into the cell, but it is the receptor hormone complex that will cause further changes and will take over the function of the protein or polypeptide hormone, and ultimately the desired and required effects are achieved. For steroid hormones like cortisol, aldosterone, estrogen, progesterone, testosterone, their receptors are located in the cytoplasm. Because these are fat soluble hormones and they can easily enter through the cell membrane into the inside of the cell. In the inside of the cell, their receptors are located in the cytoplasm, and the binding of the hormone with the receptor and the hormone receptor complex will cause further changes in the nucleus, and it will cause the expression of the uh, genes that are responsible for the synthesis of structural and functional proteins that will bring about the desired or required effect. For tyrosine derivatives, the receptors are located in the nucleus. For example, the receptor for the thyroid hormones T4, T3, they are located in the nucleus, and the thyroid hormone binds to this receptor located in the nucleus to cause further changes and to bring about the required effects. So, the lipid soluble hormones, for example, steroid hormones, they have got their receptors inside the cell. Steroid hormones have their receptors in the cytoplasm, and thyroid hormones have got uh, their receptors in the nucleus. And the hormone receptor complex, once it is formed, it will stimulate certain promoter and enhancer regions in the DNA and will cause the expression of specific genes that will synthesize structural and functional proteins, and the desired effects are achieved. On the other hand, for the peptide hormones like anterior pituitary hormones, posterior pituitary hormones, and hormones secreted by the pancreas like insulin and glucagon, they and parathyroid hormone, they actually doesn't enter into the cell, but they bind to the receptors like G coupled protein receptors on the cell surface. And once they bind to the receptor changes its structure. And this binding of the peptide hormone to the receptor will activate the second messenger systems. For example, cyclic AMP and mediators from the membrane phospholipids and various channels like ionized calcium channels. So this is the second messenger system function and its activation that will cause the nucleus to express certain specific genes to achieve the desired effects. The disorders of the endocrine system may take one or two forms, and the problems are either caused by the presence of the mass lesion. For example, when an endocrine gland, if there is a tumor, it will cause the gland to enlarge in size. Sometimes inflammation may also cause the mass of the endocrine gland to enlarge, and the enlarged mass of the endocrine gland will compress the nearby organs, and here the effects are totally produced by the presence of the enlarged mass of the endocrine gland. For example, if the pituitary gland enlarges, it will compress the bony space known as the uh, sella turcica, where it is located. Similarly, the enlargement of the thyroid gland will push the esophagus and trachea as they are the nearby structures. So these effects are known as the mass lesions. Now, the abnormal endocrine gland may be either functional, it will be secreting excess, or it may be causing overproduction of the hormone or underproduction of the hormones, hypo and hyper, or it may be non-functional. If it is not functional or non-functional, the effects will be simply the morphological changes and the mass lesions that are due to the physical presence of the, for example, a tumor in the gland that is compressing the nearby structures. But if it is functional, it will have both effects. Similarly, on the other hand, the endocrine gland may not have mass effects as it doesn't get enlarged and doesn't get to develop a mass, and simply the effects are either because of the overproduction or underproduction of the hormones. Now, the underproduction or overproduction of the hormones will lead to biochemical and clinical consequences. Now, when we investigate an endocrine disorders, we look both for the presence of the mass and we also look for the functionality of the endocrine gland. To see that either there is an overproduction of the hormone or underproduction of the hormone, we will measure the level of the hormones. Also, we will measure their regulators and other metabolites or end products of the hormones to make an assessment of the level of the hormone in the serum. For mass effects, we will use various techniques like imaging techniques, X-ray, ultrasound, CT, MRI, and by combining the two investigations in one where we measure the level of the hormones and in other when we see for the uh, mass present due to the enlargement of the endocrine glands, for example, and then we will go to some interpretation to diagnose the endocrine disorders, and this will lead to guide us to the therapy. So, endocrine disorders may arise either because of impaired synthesis of the hormones, and sometimes the hormone is synthesized normally but it is not released into the blood, and it may also be due to abnormal interaction of the hormones on the target organs and target tissues. Whatsoever is, but the result will be an abnormal tissue response. Now, to measure the hormone levels in the blood, it is very important, and here in the lab we use various techniques. For example, we use the enzyme linked immunosorbent assay. We also use radio immunoassay and other techniques to measure the level of the hormones in the blood. Now, here in this slide, we discuss the ELISA, which is the enzyme linked immunosorbent assay, and without going into the very minute details, I will simply explain the mechanism, the basic principle by which hormone level is measured in the blood. We have this microtiter plate which is used in ELISA, and it usually contains 96 wells. Now here we take one well, and the surface, the inner surface of this well is already coated with antibodies for a specific hormone. Now if we want to measure the level of that specific hormone, we will add the serum that we suspect to contain that particular hormones, and we know that antibodies are already present in the inner surface and bottom of the well. Now the hormone will directly go and it will tightly bound to the antibody. Now we will discard the remaining of the serum, as we know that the hormones present, they are tightly bound to the antibodies and they will not be washed away. Then we add another antibody which carries an enzyme linked to this antibody. And what happens here, that the enzymes that is linked to the second antibody, the quality of converting a substrate from a non-color or colorless substance into a colorful substance. Now, if the hormones was present, that would bind tightly to the antibody, and this antibody will go and it will bind to the hormone. Now, here in the third step, we add the substrate. If the hormone was present, the enzyme linked antibody would have bound to the hormone, and this enzyme will convert this substrate through a chemical reaction, and it will be converted into a colored substance. Now, once the substrate is changed into a colored substance, we use a colorimeter to measure the change in the color. If it is a dilute form of change, then this means that the hormone concentration was comparatively less. But as it becomes concentrated, it means that the hormone level in the serum in question was higher. And the colorimeter or photocalorimeter, it is bound to an automated computer, and the computer will tell us it will measure the change in the color, and it will calculate it, and it will give us the level of the hormones in the blood. Here you can see that initially the antibodies are attached previously to the will for the known hormone. Then we add the hormone, and if the hormone gets the antibodies, if the hormone is present here, it will bind to the antibodies, forming the hormone antibody complex. In the third step, we add another antibody specific for the hormones that is linked to an enzyme. And the hormone was, if it was present in the serum, this antibody will bind to the hormone, the second antibody. And since it contains the enzyme, then we add a substrate, and we know that this enzyme will change the color of the substrate, and we will measure this change in color by using the colorimeter, photocalorimeter, and that is connected to a computer that will calculate and will give us the level of the required hormone. Another method which is very common for the measurement of the hormone levels in the blood is the radioimmunoassay. In radioimmunoassay, the radioactivity of the solution is measured. And what we do, that we first take a known concentration of the hormone. We know here that how much hormone we have added. We know the concentration, and this purified standard hormone is tagged with radioactive isotope. So what we do, that since we know the concentration of the hormone, so this is standard for us. And when we measure the radioactivity of this standard hormone, by this we can calculate the hormone in question. That is not known to us. Now, since we know the radioactivity of the standard, so we can find out by the radioactivity of the sample, and from this radioactivity we can measure the level or concentration of the hormone in the serum. This is very important graph, and here we first make the standard. We take, for example, 2 milligram of the hormone, and we measure the radioactivity. Suppose it is 20. When we add 4 milligram of the hormone to the solution in question, the radioactivity is 40. And in this way, we make this chart, and we come to know that 10 milligram will give us 100% radioactivity. Now what we do in the third step, that we have this plotted graph. Now we take the hormone in question that we want to measure its concentration, and we simply measure the radioactivity of the hormone in question, and then we put the radioactivity of the hormone measured in question here, and we can calculate, and we now, by using this graph, we can directly know the concentration of the hormone in the serum. For example, we know that for 40 radioactivity, the concentration will be 4 milligram. So if this unknown hormone give us 40 radioactivity reading percentage, then we can easily put this here, and we know that the hormone concentration will be 4 milligram per deciliter. Thank you very much.